much for this introduction. My name is Andreas Dräger. I work at the Center for Bioinformatics in Tübingen, and here I would like to show you a tool that we have implemented together with the University of California, San Diego, the Systems Biology Research Group. It's called Escher. It runs in the web, and on this slide you already see some nice teasers of what it can do, namely drawing pathway maps and visualizing all kinds of omics data on it. So mainly, Escher has three key features that you can use. So the first one is there's a builder tool that allows you to rapidly design your pathway maps. So it has all kinds of search functions. You can upload a model and then search what is in that model based on gene names, reaction names, or metabolite names, and it will pop up with these boxes, and then you can say I want this reaction here, and then it draws the reaction arrow with all components participating in it, and it has some built-in linear tools so that you can quickly build a linear pathway in all kinds of directions, and once you have that, then you can combine them here and easily make a connection between them, and so then you get your first pathway model ready. And then you can, of course, modify everything afterwards using Bezier curves and everything. So then once you're happy and you have introduced some nice circles here and everything, so that it looks like a real textbook image of a pathway, then you can go ahead and visualize data on it. And here in these panels, you can see that we have multiple sorts of data that we support. So there's, for instance, flux data that you can upload here. And then you get the reaction velocity visualized, or you can indicate metabolite concentrations here, and then visualize that. Or you can even use transcriptomics data, because we have built-in gene-protein reaction rules, so that you can see how do transcriptomics data influence or impact my reaction network, my metabolic network. Of course, everything is customizable. There are numerous settings, so you can change the color scheme and everything, gradients. It's all available as you like. And then finally, when you're done with that, then you can share your maps in different formats. So Escher has a built-in format that is based on JSON that it understands directly, and this can be exported to the SVG format, which is a vector graphics format that is supported by most current browsers. And so everything, including the data visualization, can directly be exported to SVG. And then we implemented a separate tool as a standalone application that you can see here on this panel, which is called Escher Converter. And Escher Converter can also interact with these community standard formats, SBG NML and SBML, and it bidirectionally can convert maps from there, so import or export, and so this can be very useful. And Escher Converter itself has a different visual style based on the systems biology graphical notation that you can see here. So there are multiple ways of indicating reaction networks. And just to show that it really scales up very well, here's the largest metabolic map that I'm aware of that is supported and runs in Escher, which is the human reconstruction, the recon map that has recently been published last year, or by the end of last year, and it contains more than 5,500 reactions, and the web tool Escher can visualize that and can interact with that, so it really scales, and it runs on a regular web browser, on a regular laptop where I did this image, so I'm not aware of any restrictions until now. So if you got interested in that and like it, then there are multiple ways of get in touch with us. So of course there's here the poster that you can come and just directly talk to me, or you can directly try Escher online. So just go to escher.github.io and you can have access directly to all functionality that I just demonstrated here. Escher Converter as a separate tool is also open source, is implemented in Java. You can go to github/sbrg for Systems Biology Research Group and download Escher Converter from there. Or we also have a YouTube channel that you can visit here at tinyurl.com/sysbio-channel, and there you can find some interesting animation videos that give you a broader overview about other activities that we also do on top of Escher. So I think there's even a little time for one question or so, and thank you for your attention.
We can do one quick questions if there are any, but otherwise I will just want to wrap up because we need to go down for lunch because they tend to run out of food very quickly here. So I really think we had a very nice session, very nice talks.